Hello and welcome to this course. This course is named as Basics of Immunology. Now, myself is Dr. Indrani Sarkar. I am an assistant professor at Kalinga University, Raipur. Previously, I have done my graduation in human physiology from Siliguri College. Then I did my master's in biotechnology from University of Burdwan, West Bengal. After that, I did my PhD in bioinformatics from DBT Bioinformatics Facility, University of North Bengal. After that, I got 2+ years of postdoc experience, which I did from SACON Coimbatore and University of North Bengal. I have 7+ years of research experience where I got 30 peer-reviewed papers with an cumulative impact factor of 114, and I have 2+ years of teaching experiences. And I am your host in this course, so let's get started. So this is the first thing, the overall view of the course, what we are going to learn in this course. So the first thing is this course will be covering the important aspects of immunology. The second is we'll start with basics of immunology and then we'll deep dive at each and every aspect of immunology. The third thing is we'll discuss the very important terms of immunology like innate immunity, acquired immunity, adaptive immunity, and also we'll discuss the production and development of B cell and T cell lymphocytes, different types of antibodies and everything, which are very important aspect of immunology. The fifth thing is the complement system, hypersensitivity, autoimmune diseases. These things are the most important part of immunology, where we get the clinical aspects of immunology and where we get the real-time application of immunology. Okay, the sixth and the last phase will be covered up by all the immunotechniques and all the clinical practices that are done in immunology. Okay, so let's start with the module first. In the first module, we'll discuss about the immune system, what are the components of the immune system, then the barrier against all these infections, innate and acquired immune system, active and passive immunity, and antigen. So this will be the part of module 1. In module 2, we'll deal with the antibody, the structure of antibody, the types of antibody, what are all these antigen antibody interactions, what are the different types of antigen antibody interaction, and we'll discuss one specific interaction that is called ELISA, which is very important, and the full form is enzyme linked immunosorbent assay. Now there are 4 different types of ELISA: direct, indirect, sandwich, and competitive. We'll do one by one. Okay. In the third module, we'll discuss about 2 main things. One is hypersensitivity reaction, which can be divided into 4 parts, type 1, 2, 3, and 4. And the second thing is complement cascade pathway. And there are 3 pathways through which this complement fixation is done. One is classical pathway, another one is alternative pathway, and the third one is lectin pathway. Okay. So in this module, we'll start with module 1. So first thing is immunology. Immunology, what this term immunology means. This is basically the study of our immune system. And what is our immune system? Our immune system is nothing but a complex network of different cells, tissues, and organs that can protect our body from foreign intruders. And what are these foreign intruders? They may be bacteria, they may be virus, some parasites, some fungus, or some chemical agents like pollutants. Okay, so the primary function of our immune system is to degrade or neutralize this kind of foreign intruders, and they did this very efficiently by distinguishing our self cells and those foreign cells. So our cells are self cells for our immune system, and those foreign particles are non-self cell for this immune system. These 2 terms, self cells and non-self cells, will use it in immunology in future. So one very interesting thing is fever a disease? So often we say we are suffering from fever, but we should know that fever is not a disease. Fever is just our immune response. Something or some foreign particle has entered into our body, and our immune system is responding against that antigen or foreign particle. That's why our body temperature raises. Because if the body temperature rises, then that pathogen cannot prevail in that high temperature. Also, this high temperature helps in the repairing mechanism of our body. Okay, so these are some components of immune system. You can see over here, this is the lymphocyte and this is the monocyte. Both lymphocyte and monocytes are part of types of white blood cells. Again, from B lymphocytes, antibodies are produced. This is again very important part of immune system. Then we have this lymphatic system, we have thymus, which is present in this part of our system, we have spleen, we have bone marrow, and most importantly we have skin. So skin is very important. This is the primary barrier of our for our body, because the skin covers most of the parts of our body, and skin has huge, the greatest surface area of our body. So this is the largest organ of our body, and skin protects us from all the foreign particles, from several bacteria, viruses and whatnot. Sometimes the dried cells are peeled off, so that is the mechanism by which all those pollutants and bacteria and parasites, they are washed off from our body. So let's deep dive into the barriers against infection. So there are 4 different types of barriers. One is physical barrier, second one is physiological barrier, third one is chemical barrier, and fourth one is biological barrier. So first one is physical barrier. As I have already mentioned, that skin is the most important physical barrier. Then we have mucous membrane, that is present in our respiratory tract, in our gut system, that means gastrointestinal tract, and in our urinary tract. The third thing which is very important in this is this part. This is mucociliary escalator. This is present in this part of our system, that means in the respiratory canal, and they contain some ciliary columnar epithelial cell. You can see over here ciliated columnar epithelial cell, and they contain cilia. So cilia, they have a flagellar movement, and due to that flagellar movement, small particles like dust and everything, they get washed off. So due to that ciliary movement, we get cough, we get sneezing. All these reflexes are due to that only. So with coughing and sneezing, let's go to this physiological barrier. So these are the physiological barriers. These are basically the processes through which we can wash away, or we can remove the foreign particles or the pathogens from our body. So it may be diarrhea, it may be vomiting, it may be coughing, or it may be sneezing. These are all physiological barriers. Now let's come to this chemical barriers. See, our skin has pH of 5.5. Our gastric acid pH is 1 to 3. The range is 1 to 3. Our vagina, it contains the pH of 4.4. All these pH are acidic, because in this acidic environment, pathogens cannot grow. Also in this lysozymes are mainly present. These are basically enzymes. These are present in these orifices like our eye and in saliva. All these things contain lysozyme, because they can degrade, they can digest the proteins which are present in bacteria, virus, parasites and all. Also some epithelial cells, they secrete a specific type of protein. These are called defensin, and this defensin protein, they have specific role against some fungus or against some bacteria. So these are the chemical barriers. The fourth one is biological barriers. The biological barrier is present within our body, and they are none other than all the good bacteria that we contain. So non-pathogenic good bacteria are there in our nasopharynx, in our mouth, throat, in our GI tract, in our vagina, everywhere. So that human microbiome, that good bacteria are protecting us from the outsider bad bacteria. So now let's come to these 2 types of immune system. One is innate immune system, another one is acquired immune system. The innate immune system is basically the first line of defense. This is non-specific, but this is very immediate. So this kind of what is the main function of this innate immune system? They can recruit different immune cells, and they can recruit different kind of chemical modulators like cytokinins. Okay, they can activate the complement cascade mechanism. They can identify the foreign particles, and they can remove those foreign particles from blood and lymph node and various organs of the body. They also help in the production of this acquired immunity that is via antigen presentation. This acquired immunity or adaptive immunity works. So that acquired immunity is being is being backed up by this innate immunity. So what are the components of innate immunity? Like physical barriers, like our skin, mucous membrane, everything, inflammation, some complement system, and our white blood cell. But some white blood cells are again a part of acquired immunity. So white blood cells like macrophages or monocytes, neutrophil, basophil, eosinophil, natural killer cells, all these are under innate immune system. Now let's deep dive into the acquired immune system. Now, acquired immune system is something which is not present with birth. It is learned. And what is this learning process? We get exposure to some foreign antigen and some antibodies are produced in our body. So that is the learning process for our immune system. That is acquired immunity. Okay. So this acquired immunity, first to develop this acquired immunity, it takes time. But what it does is, whenever we are exposed to a foreign particle, it produces some memory cells, and that means whenever there is subsequent second exposure of the same antigen, it will work specifically against that antigen very quickly. So that is the thumbs up point for acquired immunity. And for acquired immunity, we have two specific cells. One is T lymphocyte, another one is B lymphocyte. Other components are like dendritic cells, cytokinins, and complement system. Everything is there in acquired immunity. Okay, so this acquired immunity can again be divided into two parts. One is active immunity, another one is passive immunity. Active immunity is this kind of immunity can be developed by getting exposure to some antigen. Like it can be of two types. One is natural antigen. That means antibodies are being produced when we are naturally exposed to some antigen. Another one is artificial antigen. That means artificially acquired active immunity. That means we are getting exposure to some antigens artificially. For example, vaccination. That is the most important example, the best example for artificially acquired active immunity. Then we have passive immunity. In passive immunity also, passive immunity means some kind of immunity we acquired from someone. Okay, like they are again, this passive immunity can again be divided into two. One is natural, another one is artificial. For natural, the example is mother's breast milk. So all the fetus, all of us, we got good amount of IgG, that is immunoglobulin G, from the mother's milk, that is colostrum. So that is the source of natural passive immunity. And artificial passive immunity, now there are options of serum medicines. So all this immunoserum medicine, that will give us artificial passive immunity. Now we have already discussed about these foreign particles. In immunology, we call these foreign particles as antigen. So antigens are those agents that are foreign to our body and that can cause some complications in our immune system, so that our immune system can react against those antigen. But there are some specific properties from this antigen, like they should induce our immune system. This is very important. The second thing is their molecular mass should be of 14,000 to 6 lakh Dalton. Then they are mainly proteins and polysaccharide, but proteins, mainly these antigens are proteins only. They are chemically very complex. The more complex they become, the more immunogenic effect they can show. Antigens are species specific. That means whatever is antigen for our system may not be an antigen for rabbit or something else. The second one is the age influences immunogenicity. That means very old people or very newborn infants, they exhibit very low amount of immunogenicity. So this is the first module. Thank you very much. We'll discuss all about antibody in our second module. Thank you.